The process of sperm production, also known as spermatogenesis, is one of the most intricate and remarkable biological functions in the human body. It is a continuous and highly regulated sequence of events that takes place in the male reproductive organs, ensuring the potential for reproduction throughout a man's life. Every day, millions of new sperm are created, a feat that requires precise coordination between hormones, specialized cells, and various parts of the male reproductive system. At the core of this process are the testes, two oval-shaped organs housed in the scrotum, located just outside the body. The positioning of the testes is no accident. Sperm production requires a temperature slightly lower than the internal body temperature, and the scrotum helps to maintain this delicate balance. Within each testis are tightly coiled tubes known as seminiferous tubules, stretching for over half a mile if unraveled, and it's here that the journey of sperm production truly begins. Everything starts with a special type of cell called a spermatogonium. These are stem cells, meaning they have the unique ability to divide and either produce more stem cells or begin the transformation into mature sperm cells. Spermatogonia lie nestled along the outer edge of the seminiferous tubules, protected and nurtured by another type of cell, the Sertoli cell. Sertoli cells are sometimes called nurse cells because they provide structural and nutritional support throughout the complex process ahead. Spermatogenesis can be broken down into three major phases: the mitotic phase, the meiotic phase, and the spermiogenesis phase. The process begins with mitosis, a type of cell division where a spermatogonium splits to produce two identical cells, one that remains a stem cell to keep the cycle going, and one that will become a primary spermatocyte. Next comes meiosis, a specialized form of cell division that reduces the chromosome number by half. The primary spermatocyte, which originally contains 46 chromosomes, divides to form two secondary spermatocytes, each with 23 chromosomes. These secondary spermatocytes then quickly undergo a second round of division, producing a total of 4 spermatids, all with just 23 chromosomes. This reduction is vital, because when a sperm cell fuses with an egg during fertilization, the resulting embryo will have the correct number of chromosomes. Once spermatids are formed, the process of spermiogenesis begins. During this phase, the round, immature spermatids undergo dramatic changes in shape and structure. They elongate and develop a tail or flagellum, essential for motility. The midpiece of each sperm cell becomes packed with mitochondria, tiny organelles that supply the energy required for swimming. The head of the sperm houses the nucleus, which contains the genetic material and is capped by the acrosome, a specialized structure filled with enzymes needed to penetrate the outer layer of the egg. Throughout spermatogenesis, Sertoli cells continue to play a crucial role. They not only provide nutrients, but also secrete signaling molecules and form a protective barrier around developing sperm, shielding them from harmful substances and immune system attacks. At the same time, Leydig cells, located in the tissue between the seminiferous tubules, produce testosterone in response to hormonal signals. Testosterone is the primary male sex hormone, and it's indispensable for the maintenance and regulation of spermatogenesis. The entire process of creating mature sperm from spermatogonia takes about 64 to 72 days. However, because new sperm are constantly being produced in overlapping cycles, the testes can release millions of sperm every single day. Once mature, sperm are released from the Sertoli cells into the lumen of the seminiferous tubules, a process called spermiation. But the journey isn't over yet. From the seminiferous tubules, sperm travel to the epididymis, a long coiled tube attached to the back of each testis. Here, sperm undergo further maturation, acquiring the ability to swim and fertilize an egg. This maturation process takes about 2 weeks. The epididymis also serves as a storage facility, holding sperm until ejaculation. When the time comes for ejaculation, sperm are propelled from the epididymis through the vas deferens, a muscular tube that transports sperm upward into the pelvic cavity. Along the way, sperm mix with fluids from the seminal vesicles, prostate gland, and bulbourethral glands. These fluids provide nutrients, protect sperm from the acidic environment of the female reproductive tract, and help form semen, the medium through which sperm travel. The hormonal control of spermatogenesis involves a finely tuned feedback system known as the hypothalamic pituitary gonadal axis. It starts in the brain, where the hypothalamus releases gonadotropin-releasing hormone, GnRH. This, in turn, stimulates the pituitary gland to secrete two hormones, luteinizing hormone, LH, and follicle-stimulating hormone, FSH. LH prompts the Leydig cells to produce testosterone, while FSH acts on the Sertoli cells to support the development of sperm. Together, these hormones ensure that sperm production continues efficiently throughout a man's reproductive years. Various factors can influence the rate and quality of sperm production. Age, overall health, diet, exposure to toxins, stress, and lifestyle choices like smoking or excessive alcohol consumption can all have an impact. Maintaining a healthy lifestyle can help optimize sperm production and quality. In summary, sperm production is an ongoing, dynamic process that requires the collaboration of many different cells, tissues, and hormones. From the division of stem cells to the maturation of motile sperm, every step is carefully orchestrated to ensure the possibility of creating new life. The body's ability to produce millions of sperm every day is a testament to the complexity and efficiency of human biology, a remarkable cycle that continues largely unnoticed until the moment it is needed for reproduction.